Hi, hello. Welcome to Practically We Bring Learning Alive. I am Dr. Preeti. Today we are going to start an interesting series for you all, which will be dealing with evolution. We will be studying evolution in short parts, where we will be taking a journey through evolution from microorganisms to human beings. It will be a short journey, but it will be an interesting journey for sure. So let's start this journey, evolution micro to macro. And to begin our session with, today we'll be studying or seeing about prokaryotes, especially about Archaebacteria. So let's begin. What do you see in this glass of water? Do you see other than anything about water? Let's find out. Well, you will need more than a magnifying glass to understand that. There were some creatures, weren't there? Well, these creatures are small, and these creatures are nothing but microbes. Yes, you heard it right. They are microbes, a short form for microorganisms. Hi, hello Manish. Good evening. So, welcome to Practically. Let's begin our journey. So, microbes are nothing but small organisms which are found in not only water but everywhere around us. So, you might be thinking how these microbes are related to evolution. Well, we will be seeing that and coming to that part in the later part of our series. To begin with, we will just understand what are the properties of microbes and prokaryotes in general. You might be thinking again that is there any type of microbes? Are there any different types of microbe? Yes, Manish, can you answer? Do you think there are types of microbes? Well, I'll tell you, microbes belong to the category of prokaryotes. Prokaryotes are nothing but organisms which do not have proper nucleus. They are a very primitive or primordial organisms or beings which are present on earth right from the very early on of the journey of the evolution. So you can say that prokaryotes were the first beings on the earth. But how they came to be, we will study that in our third session. So let's find out some of the basic properties that belong to prokaryotes. Yes? So they are small and mostly single-celled organisms. So they are small and single-celled. So what you will require to observe them? It's a general question. Can you answer, Manish? What will you require to observe prokaryotes? Well, we will require a microscope. Isn't it? A magnifying glass won't be able to capture the structure of microbes because these microorganisms are very small. Small as in they are less than millimeter. They belong to 1 to 1.5 micrometer. And micrometer is a very small scale which is 1,000 times less than 1 millimeter. Can you believe that? These organisms are such tiny beings and they can be found everywhere. The prokaryotic cell, now we will be seeing about the structure. Well, the prokaryotic cell is surrounded by plasma membrane, but they lack internal membrane bound organelle. Do you understand what that means? It means that they don't have any membrane bound organelles. Like in humans or plants, we have mitochondria, chloroplast, lysosomes, etc., which are present in our cells. But prokaryotes do not contain these type of organelles. Instead, they have genetic material. Okay, they have genetic material, that is, they have DNA, which is present in the center. Okay, DNA which is present in the center, and it is called as nucleoid because the DNA material doesn't have any nuclear membrane. It doesn't have any nuclear membrane. Well, Manish, my name is Dr. Preeti. You can just call me ma'am. Okay, so absence of nucleus in eukaryotic cells, that is in human beings or any animal cells, you might have seen that we have a membrane bound nucleus containing a genetic code. But in case of prokaryotes, they lack the nucleus, absence of nucleus, and they have only nucleoid present, which is nothing but a nuclear, or that is genetic material or DNA in the center of the cell. They also contain additional DNA. Now this information is very important because this is what makes them apart from the eukaryotic cell. We don't contain extra DNA material. We only have our genetic material in the form of chromosomes. But in case of prokaryotes, they have additional DNA in the form of plasmids. Plasmids are nothing but small circular double helical DNA. And this helps in the protection of microorganisms or prokaryotes. So keep this information in mind. Prokaryotes are basically divided into Archaebacteria and bacteria. Bacteria are also known as eubacteria. Why? Because eubacteria are the true form of bacteria, whereas Archaebacteria are a very primitive form of bacteria which were supposed to be present right from the beginning of Earth. But later on it came to know that they evolved, or they came to life when the origin of life occurred on Earth, which we will be studying that in the third session. So now let's see the structure of prokaryote. This is the general structure. Okay, this is the general structure because this is similar to eubacteria. That means it is similar to Archaebacteria as well as bacteria. So what does this contain? It contains the plasmid DNA. As I said earlier, it contains the plasmid DNA. It is a double-stranded DNA in a circular form. It can be one, it can be many. It gives the property of protection. Okay, protection against the environment or any harmful antibacterial agent which we can give them. Then they have cytoplasm. They have ribosome in the cytoplasm. Right? They have pili. Pili are nothing but small hair-like structures which help them in motility along with the flagellum. Flagellum is nothing but a long tail which helps the organisms to move. Okay. Then they have capsule. Capsule also prevents the cytoplasm from oozing out, and they have a thick cell wall. They have plasma membrane which contains lipid molecules. Now let's see some of the properties of Archaebacteria. This is the Archaebacteria which you can see, and this is a electron microscopic image. Well, as I said earlier, you can observe microorganisms through microscope. But to have a better understanding of shape and size, we have something called as scanning electron microscope. Okay, and this microscope is a very advanced type of microscope which can observe the organism in actual sense. You can see, right? And they also give us the measurement. This organism is nearly 1 to 2 micrometer in size. It's very small, and with the help of regular compound microscope, you cannot observe the details of structure. Hence, scanning electron microscope is required. Now let's see the properties. Archaebacteria, as I said, belong to the prokaryotic family. They are obligate or facultative anaerobes, which means they are either compulsorily anaerobe. Anaerobe means they do not need, no need of oxygen. Yes, they can survive without the oxygen. Hence, they can be observed in extremes of environment. Extremes of environment means they can be observed in hot springs. The temperatures are beyond 100 degrees Celsius. Can you imagine the area? We can't even live in 32 or 35 degrees Celsius, isn't it? So just imagine the organism living beyond 100 degrees Celsius. Amazing. Then they live in deep oceans. In deep oceans, the environment is such that sometimes there are deep hydro vents, which again have high temperature. But as we go deeper into the ocean, we can see that there is a high pressure, and high pressure can kill us instantly, isn't it? But these organisms survive the high pressure zone. They can even be found in glaciers. Glaciers means they can be found in Arctic and Antarctic. The regions are extremely cold, yet the organisms are thriving over there. And because of this, they are labeled as extremophiles. Extremophiles. That means they love to live in the extreme of condition. Philus means loving. Loving. So they are loving the environment, extreme environments to live. They have rigid cell wall. As you can see that the environments that they live in is extreme. That means they have to have some protection against them, isn't it? So they have rigid cell wall that protects them in the harsh environment. Then their cell membrane consists of lipid layer. Okay, and lipid layer helps them to survive or helps them to have some flexibility. Okay, and because of that they can also survive the cryogenic conditions. Cryogenic means they can survive the cold temperature. They do not possess membrane-bound organelles. As we have established earlier, Archaebacteria, being a prokaryote, doesn't have any nuclear or any membrane-bound organelles in them. Then their mode of reproduction is asexual. It can be binary fission, it can be fragmentation, isn't it? So they reproduce asexually. So these are the properties. I hope you like this session. Now we will be seeing some of the examples of extremophiles. Yeah. So thermophiles. Thermophiles means heat loving. Heat or temperature. Can you guess this image? Anyone? Can you guess this image? This is from Yellowstone National Park. Yellowstone National Park is a famously known place for hot springs, and it contains a volcanic water lakes where water is boiled through the volcano which is active in the bottom of the ground. So this Yellowstone National Park is a hot zone famously known for having Archaebacteria present in the vicinity. And the organisms, example of an organism that I have given is Sulfolobus metallicus. I think you might not be able to pronounce the name easily, but based on the property or based on where they are living, they are labeled as. Sulfolobus means they are sulfur loving. Metallicus means they also have some metallic loving properties. And these organisms are usually present in the high temperature zones. Then we have Piezo Files. Piezo Files are high temperature loving, high pressure loving. Yeah. So high pressure loving organisms. Example is Thermococcus piezophilus. This organism can be found in hydrothermal vents. Okay. Hydrothermal vents which are deep in the ocean. Yeah, deep in the ocean. That means they also have high temperature and a very high pressure. And these organisms thrive in such condition. Then we have acidophilus. Acidophiles are acid-loving organisms. Okay, acid-loving organisms means they can survive in high acidic conditions. Now, acidic condition means, I suppose you might know that we have an acid range of 1 to 14, right? So, as we go from 14 to 1, acidic condition increases and it becomes extremely acidic. We cannot survive. We will burn our fingers and ourselves if we touch a acid which have 1 as a pH. Okay. But if we go from 1 to 14, the pH property increases and that becomes basic. So basic means alkaline, and these organisms are also present in alkaline conditions known as halophiles. These halophiles are salt-loving or alkaline-loving organisms, and they are present in extreme high salt concentration regions. So now, from these examples, you might have understood that Archaebacteria are extremely environment-loving organisms. If you know few more regions, just name them in the comment section so that we will also come to know what you know about extremophiles. So in this session, we are only going to cover Archaebacteria as such and briefly that too. So in the next session, we will be covering bacteria, which are also known as eubacteria or true form of bacteria. So do join us on the second session, which is going to be aired tomorrow at 5 p.m. For more such interesting videos and content, like the Practically Life channel on YouTube and subscribe our channel. And don't forget to hit the bell icon so that you will be updated with a notification. Download the app from Play Store and App Store for more such interesting videos and contents. See you tomorrow. Bye bye.